AS Level Biology 9700, Paper 1, Variant 2, May June 2025. Students, this is the second video on this paper, and in this video, we will discuss the question number 11 to question number 20. The question number 11 is which bonds are found in the levels of the structure of a protein molecule. So they have given us three structures in the table. The first one is the primary structure, second is the secondary structure, and the quaternary structure. So it means we have to find out which option is the correct option. So I would like to discuss these structures. First of all, I would like to discuss the primary structure. Primary structure, it is a linear sequence of amino acids is held together by peptide bonds. So amino acids are joined together with the help of peptide bonds. So peptide bonds, C or D can be the correct, A and B are not correct. Secondary structure. So secondary structure, uh, it consists of two types of structures, that is the alpha helical and beta plated sheets. So they are folding in this way. Alpha helical and secondly the beta plated sheets in this way. So sheet-like structure is formed. And during this formation of alpha helicals and beta sheets, hydrogen bonds are formed between the atoms in the polypeptide backbone, hydrogen bonds. So hydrogen bonds will be formed in secondary structure. Ionic bonds will not form. And when there is quaternary structure, this level involves multiple polypeptide chains, multiple polypeptide chains, or as in the formation of hemoglobin, there are four polypeptide chains. So in this way, this type of structure give a 3D structure. So here in this case, ionic bonds, hydrogen bonds, and sometimes sulfide bonds are also there. So ionic bonds. So we will go with the ionic bonds because the C option is giving the correct sequence. So C is the correct option. Question number 12. And question number 12 is prokaryotes and eukaryotes use extracellular enzymes. Extracellular digestive enzymes are found in the external environment of some prokaryotes. Some prokaryotes are digested by hydrolytic enzymes during phagocytosis by eukaryotic cells, which shows the location where extracellular enzymes are produced. Extracellular enzymes are produced, where they are produced. So I would go first of all, what are prokaryotes? So the prokaryotes are organisms which don't have a proper nucleus. So their DNA is directly embedded into the cytoplasm. So these organisms produce enzymes inside their cells and then secrete them into the external environment to break down large food molecules. So this is form of extracellular digestion. So they perform extracellular digestion, but the enzymes are produced inside the cells, which are secreted out, and then they are used for extracellular digestion. So we will go with these inside cells. And secondly, the eukaryotes. Eukaryotes, the organisms with well-defined nucleus, and these cells, as for example, like white blood cells, say they engulf foreign particles or pathogens, into a vesicle. So these vesicles then fuse with lysosome and organelle that contains hydrolytic enzymes. So the digestion of the engulfed material occurs inside the cell within their new vesicles. So if the digestion is occurring inside the cell, again, I would like to say that the enzymes will also be produced inside the cell. So we will go with the inside cell. So all other options are not correct. Only A is the correct option. Then next is amylase breaks down starch molecule. Amylase is an enzyme which breaks down starch molecule, which is a polysaccharide. So which substance will have the same number of molecules for the duration of the secretion? Amylase is an enzyme, Enzyme is a substance which acts as a biological catalyst and speeds up a chemical reaction without being used in the process or consumed in the process. It remains its quantity almost remains the same. So it means during this reaction, enzyme is that substance which remains the same. Whereas the water can be used, so its quantity maltose, it will break down, its number will increase again. Amylase, its number will also increase. So these B, C, D are not the correct option. Only A is the correct option because this is the enzyme which will remain the same throughout the reaction. Now question number 14. Question number 14 is which graph could show the effect of pH on the rate of enzyme catalyzed reactions. So they have shown us the 4 graphs. So here I would like to say uh, we have to choose 2 graphs because the answers are including 2 graphs. So enzymes function best within a specific pH range. The pH is the power of hydrogen ions. So this is every nutrient has the specific pH. So here I would like to say at the optimal pH it is 14. So 7 and 1 to 14 and optimum is 7. So according to this, at this pH, the enzyme's active site has the correct shape to bind to the substrate efficiently. So changes in the pH above or below this optimum can denature the enzyme. So according to this, we can choose this in uh, this reaction number 2 as it shows a proper shape. So on the other hand, number 1 will not show as the uh, at optimum it is showing the lowest value. So it is not the correct. Now I would go with the C. C, you can see as the reaction is increasing, rate of as the pH is increasing, the rate of reaction is increasing, but after some time it is smooth. So it does not happen. The pH is specific. It will increase or decrease if there will be change in the specific pH. So the different nutrients or the different substance behave differently at different pH levels. So they have specific pH. So if the pH is increased or decreased, so the shape will be changed. But in this case, you can say the graph number 4 is also going according to this phenomena. That is, the changes in the pH above or below this optimum can denature the enzyme. So by disrupting the bonds that maintain its 3-dimensional structure, thereby reducing or eliminating its activity. So both these 2 graphs, 2 and 4, are going with the rate of enzymes catalyzed reactions. So they are actually showing the effect of pH on the rate of enzyme catalyzed reaction. So according to this, C is the correct option. Now I'm going towards the question number 15, and question number 15 is, after a heart attack, the enzyme lactate dehydrogenase leaks into the blood plasma from damaged heart muscles. What is required to measure the activity of lactate dehydrogenase activity in a sample of blood plasma? So here I would like to say that sterilization of blood plasma by heating. If we sterilize the blood plasma or heat it for sterilization, then you can see it will denature the enzyme. Due to denaturation, it will become inactive. So we are not going with it. Now the second, incubation of sample with substrate for lactate dehydrogenase. If we incubate the sample with substrate for lactate dehydrogenase, so it will work. It will work because it will help. So it is the correct statement. The third statement is, incubation of sample with lactate dehydrogenase inhibitor. Inhibitor is that substance which will slow down or stops the enzyme's activity. So again, it will not go. We will not go with it. So according to this, the only second column statement is correct. So we will go with it, and this is the C is the correct answer. According to this, C is the correct answer. Next question. Next question has been showing the four graphs in which graph correctly shows Km and V maximum. Km and V maximum. So first of all, you know, the enzyme is a biological catalyst, substrate is a molecule that on which an enzyme acts. Or V maximum is the maximum rate of reaction when all enzyme active sites are saturated with substrate. So at this point, adding more substrate will not increase the reaction rate. And Km is the Michaelis-Menten constant. So Michaelis constant is the substrate concentration at which the reaction rate is half of the V maximum. Half of the V maximum. So a low Km indicates with enzyme affinity, high enzyme affinity for the substrate, meaning the enzyme reaches half its maximum speed at a low substrate concentration. So a high Km indicates a low affinity. So the rate of reaction, so we have to find out the a relationship between V maximum and Michaelis constant. So according to this, the graph number A and B, they are showing the concentration of enzymes. This is not the relationship between the concentration of enzymes. So this is the rate of reaction and the concentration of substrate. So graph should be between them. So, so according to this, the graph A and D, you can say this is incorrect for defining Michaelis-Menten and V maximum. So A and D are not the correct graphs. Then B, we go with the B and C. Both are, they are showing the concentration of substrates and the rate of reaction. So this graphs B and C. So, but there is a difference in B and C. The graph C is value is shown on the y-axis. Km value is shown on the, whereas the Km value is the value of the substrate concentration. So it should be on x-axis, not on y-axis. So C is not the correct option. whereas the B is the correct option, which is on x-axis. The concentration of the substrate is there, and then Km value is on the x-axis. So the curve shows a hyperbolic relationship, which is characteristic of Macaulay's mentioned constant. So this curve approaches a maximum rate. You can see this is the V maximum, and Km value can be found out, which is half of the V maximum. And this value can be seen on the concentration of the substrate and on the x-axis. We can find out it on x-axis. So B is the correct option. Now we are moving towards the question number 17, and question number 17 is cell membranes become less fluid as the temperature decreases. As the temperature decreases mean it is towards the coldness. So bacteria and yeast cannot regulate their cell temperatures. How do bacteria and yeast maintain the fluidity of their cell membranes when the temperature decreases? So they have given us three statements and we have to find out the correct one. Which one is the correct statement? The first statement is they increases the number of unsaturated fatty acids in phospholipid molecules. Well, this is the correct statement because the number of unsaturated fatty acids means they have double bond in their chains. So when there is double bond, it means they are unsaturated fatty acids. So they can easily break down. They have loose ends. So due to which they can maintain the fluidity of their cell membranes. Second statement, they increase the number of saturated fatty acids in their phospholipid molecules. If saturated fatty acids are increased, it means there is no double bond. So they will not be able to increase the fluidity. So second statement is not correct. This increases the number of cholesterol molecules in their cell membrane. So this is the ability of the, you can see the cholesterol molecules, if they are in their cell membrane, so they can increase the fluidity. So they give a fluidity to the cell surface membrane. So this is the basic function of the cholesterol as they are present in the cell membrane. According to this, statement number 1 and statement number 3 are the correct statements. So we will go with the A option, which is correct. Moving towards the question number 18, and question number 18, you can see they have shown us samples X, Y, and Z are epidermal tissues cut from an onion. Each epidermal tissue was immersed in one of three different concentrations of a salt solution for 30 minutes. A student observed each tissue sample with a light microscope. Then the student estimated the concentration of each salt solution using a scale. The diagram shows the scale P, Q, and R represent the estimated concentration of the three salt solutions. So the point P very close to the distilled water and moving towards the concentration with salt solution. So P point shows that if we will keep any cells into them, so water will enter into them and they will become turgid. Cells become turgid or swell. At Q, the water, as the concentration is increasing, so they will become hydrolyzed. Again, they are very concentrated solution. They are completely plasmolized. And here I have used the word hydrolyzed. No, I have to use the word. Mistakenly, I have used it. Plasmolized. Plasmolysis will start. Okay. Now we will move towards the three samples. And these three samples are has been shown here. You first of all the sample X. We have to find out the which sample X and Y are immersed. Y, you can see they are completely plasmolized. Sample Z, they are little bit, you can see, in normal condition. And X, we have to go with the Y and Z. And X, they are starting plasmolysis. They are starting there. So here, again, I'm moving towards which row shows the estimated. So Y, they are completely plasmolized. So it means they are at R. Y is with R, and Z, they are normal. So they are at P. And X, they have start plasmolysis. So they are at Q. So according to this, Y is at R, completely plasmolized. They are round shaped. So D is the correct option. A, B, and C are not the correct options. You know why the cells are plasmolized? So this is the actually simple process of osmosis in which the water moves from its higher water potential towards the lower water potential. As we add the salt into the water, so its potential will decrease and it will be highly concentrated, and only the distilled water is that shows the maximum potential. So it means when we keep the cells into the concentrated water with salt, so its water potential will be decreased. So it will lose water or it will be completely plasmolized. So moving towards the next question number 19, and question number 19 is the graph shows the results of an osmosis investigation using potato tissues. Again, osmosis is the process in which the water molecules move from higher water potential towards the lower water potential. So what is the concentration of sodium chloride solution that has the equivalent water potential to this water tissue, and what is correct about the movement of water at that point? So concentration of sodium solution that has the equivalent water potential. This water, when there is equivalence in the water potential, then what will happen? There will be no movement of water molecules. So what will happen if there is less water potential? So water will move from higher water potential towards the lower. And then in this way, the water movement will take place. When there is equivalence in the potential on both sides, that is in concentration of sodium chloride solution and the potato tissues, both they have equivalent potato equal concentration. They have equal NaCl solution. So it means there will be no movement of water. No movement of water. So we have to find out a place on graph where there is no movement, the positive. So these points shows, these are the positive points. So the positive points percentage change in mass will indicate the potato has gained water by osmosis. So as water is gained by a potato, so its percentage change in mass, its mass will increase. Or if it is losing water, then the percentage in mass will decrease. So when there is negative value, it means it's decreasing in mass. So we have to find out a point where there is no change in mass when there is concentration of both of them is equal. So we will find that point on graph. Actually, this is the point on this one. So this is the actual point when there is. So according to this, I'm trying without scale because my laptop is attached, so unable to keep a scale over it. So, but you have, when you are doing your work, then you must use the scale to draw these lines. So according to this, this value is less than 0.4, 0.4 and more than 0.35. So we will go with this value, which is A 0.37. So this is the correct option. Whereas the 17 and net movement of water out of the potato tissues, no, there should be the equal value. When there is equal value, there will be no movement, and this point is lies on this line 0. So 0 and no movement of water, no, it will not be 0. It will not be 0. 0 will show the minus 20 value. So is it no? 0.9, 0.9 is there. Again, there is, you can say, negative value over here. So we will go with the A option. A option is the correct option. Well, we are moving towards the question number 20, and question number 20, some agar was colored pink using a pH indicator. The pink agar was then used to make three uh, agar cubes with different dimensions. So they have given us 2 into 2 into 2, 4 into 4 into 4, and 5 into 5 into 5, and each cube has 6 spaces. So the cubes are placed in a beaker and covered with 0.1 molecular hydrochloric acid. The temperature of the experiment was standard at 20 degrees C. Hydrochloric acid diffused into the agar, causing the cubes to become colorless. So what is the surface area to volume ratio? So first of all, what is surface area to volume ratio? So we need to calculate surface area volume ratio. So for this purpose, surface area to volume ratio, we have to find this. So the surface area of a cube, surface area is equal to 6 into 6 is the 6 faces of a cube, square of the side. We will take this surface area for E, and then we will calculate the volume. So first of all, we will go with the cube to SA is to volume. So SA is 2, 6 faces, 6 into side is 2. We will take this care of, and then it will be 6 into 4. It will be 24. So it will be 24, and then it will be, we will divide the 24 with 8. So it will become 3. So surface, it will be 3 is to 1 for cube. Now we will move towards the cube 2, which has 4 centimeter space. So again, S A is to V. S A will be equal to 6 phases into 4 square. So SA is 6 into 16. It will be equal to 96, and volume will be 4 into 4 into 4 is equal to 64. So surface area is to volume is 9664. It will be equal to 1.5. So it is 1.5 is to 1. And then the third cube is go with the 5. So SA is to V. So first of all, SA is 6 into 5 square of 5. 6 into, so it will be equal to 150. And then volume will be 5 into 5 into 5. It will be equal to 125. So SA is equal is to 5. So it will be 150 is to 125. So it will be 1.2. 1.2 is to 1. So these are, we have find out these calculations, but here you can see, so we have find out these. So but the answer is, what is the surface area to volume ratio of the agar cube that become colorless in the least amount of time? So if the surface area is high, then it will take less time. It will take least amount of time to become colorless. So if we compare all of these, we will find that the least surface, highest surface area is this one. And after this, this is this one on the second number. As the size is increasing, the surface area is decreasing. As the size is increasing, the surface area is decreasing. If the surface area is high, it means there will be the least amount of time will be used to become colorless. So according to this, we will go with the D answer, which is 3 is to 1, which is 3 is to 1. So C, B, A are not correct option. C can be a correct option if they are asking which will take the maximum time, which will take the maximum time, or which will take the more time as comparison to the smaller cubes. So I would like to say, if the size is decreasing, the surface area is increasing, so it will take less amount of time to become colorless. So D is the correct answer. Well, students, it was all about from question number 11 to 20. I tried my best to explain them well. But if you find any problem, you can message me. But please don't forget to like, share, and subscribe my channel. Please also tell your other friends and class fellows about my channel. And thanks for watching.